So thanks for your invitation, for your kind invitation to this interesting Congress. And today I'm going to introduce a kind of tool for working in the study of drug delivery, specifically in the brain. So that's the in vitro blood-brain barrier model for drug brain permeability testing. So first I'm going to introduce this kind of technology that is called organ on a chip that has appeared some years ago. So it's quite young technology. And with this technology, what we are trying to do is to mimic part of an organ, or not really the whole organ, but at least the functionality of this organ inside a chip. So we microengineering the shape of the part of this organ, and we integrate microfluidics. We integrate also 3D co-culturing of cells for mimicking the functionality of this organ, and also sensors for the monitoring of the evolution of this chip. So from this last year has been developed different kind of organ on a chips for different applications. So for example, I think you can see my pointer. Okay, so for example, in the case of lung, has been developed and published the mimicking of the alveolar membrane of the lung, and can be detected oxygen when it's passing through the membrane. Also, it has been highly reported the mimicking of the functionality of a heart with myocardial cells breathing, so trying to beating inside the chip, and different kind of applications as a spleen, as we reported some years ago. And nowadays we are working in brain. That is the one that I'm going to show you. But yeah, as you can see, there are many different systems reproduce it in these organ on a chip systems. The main applications for these organ on a chip platforms is mainly to have easy method for reproducing the functionality of an organ, mainly for mimicking a disease. So in a chip, you can control quite well the functionality of this organ, and you can characterize it easily than in vivo. And obviously also for testing different therapies for these diseases. So with this kind of technologies has been highly starting to be used for drug discovery, for analyzing the concentration times profiles of drug absorption on this part of the tissue that is under a study. So basically, it's an innovative technology that permits the development of drug discovery in a faster way. So comparing with in vivo models that are the current technologies for drug discovery, in the case of animal testing, the advantages is that in the animal we represent the complexity of the whole organ with more complex environment. However, this kind of technology is ethically controversial. Many people are against the use of animals, but probably the most important is that are not really mimicking perfectly the organ of a human, because the cell behavior of a human is not exactly as an animal, or even with developed animals, not like rats, but also in this case are not behaving in the pathophysiology in the same way. So, and also are obviously high costly and labor intensive, not just for the animal as itself, but also for the maintenance of this kind of laboratories, for the training of the people. So it's quite costly and difficult to work. In the other way, in the systems, in the organ-on-chip systems that are in vitro, we can use human cells, so we can mimic more closely the mechanics of the cells, because we are using human cells, and even we can use also with human pluripotent stem cells. We can even use the cells of the patient for mimicking better the disease, for making a kind of personalizing mimicking of the disease, and more reproducible, easy to analyze, and easy to fabricate, and more cost effective for drug discovery and also for study the disease. And also it's important to know that 9 to 10 drugs that has been developed with the current technologies are not really reaching humans because are not or are toxic or are not effective. So it's necessary to make a change in this kind of technology. So if we compare the different technologies that we are having currently in drug discovery, we can summarize it in this graph where we are plotting the physiological relevance and complexity of the model, and also the reproducibility, so the easy to work and the reproducibility of these systems. So obviously in the physiological relevance we have the in vivo systems, and a little bit with lower relevance the 3D cell culturing. But also comparing with the 2D cell culturing that is less relevant but are easy to make it more reproducible, we have the evolution of the combination of these two technologies, that is the 2D cultures modified to the 3D cultures and evolved to the organ-on-a-chip systems, that we have the 3D culturing inside a chip, and we are also using other advantages as the shape, as the microfluidics, and as the sensing. So if we look in the drug discovery stages from the disease identification to the therapeutic, to define which is the therapeutic target, we can also start in the therapeutic target to use organ-on-a-chip systems, because are usually working with 2D culturing of cells that are not so close to mimicking the real behavior. And the main step where we can introduce advantages with the organ-on-a-chips is in the lead identification, so we can make a high throughput analysis of the different potential drugs, and we can make a high screening make a real and efficient analysis of the toxicity of these drugs and the efficiency of these drugs. So we cannot move we can move with lower potential drugs in the preclinical, saving a lot of cost and saving a lot of animals. So we are not substituting completely the use of animals, at least at this point, but at least we are reducing a lot the number of these animals and the cost of this development. So looking in the different phases, we are affecting in these stages in the drug discovery, and we can save some hundreds of animals at this stage. If we look in the per cycle of emerging technologies, we are in a very exponential growth of the expectations of this technology. Are relatively young technologies that are growing and are evolving with more complexity, and has a lot of expectations in a future, but because could be the key technology for drug discovery. Okay, so I'm going to present the technology that we are developing in our laboratory at Institute for Bioengineering of Catalonia in Spain, in Barcelona concretely. And we are working in providing an organ-on-a-chip for mimicking the blood-brain barrier, for analyzing the permeability of different nanotherapeutics agents for neurodegenerative diseases. So the first step in developing an organ-on-a-chip platform is to understand very well which is the organ that we are trying to reproduce. So the blood-brain barrier is the most highly selective semi-permeable barrier that we have into the body. So it's a large network of blood vessels that we have in the brain that are used for bringing the nutrients and the oxygen to the millions of neurons that we have into the brain. In fact, the brain is the organ that consumes more oxygen and glucose in our body. So it's highly important, the functionality, the well functionality of this organ, of the blood-brain barrier, but also has a very relevant function, that is to prevent the entrance of any toxic pathogen into the brain. So it has a very two important roles to bring all the nutrients to the brain and to prevent the pass of the pathologies and toxics into the brain. So looking with more detail to this blood-brain barrier, to this complex network of vessels, we look in the cellular structure of this blood-brain barrier. We can find endothelial cells here in purple, making this structure of the vessel, this circular vessel. That is the same kind of cells that we can find in any vessel in the rest of the body. But in the case of the brain, they are surrounded by these perocytes and astrocytes that are molecules that help on the productions of tight junctions, that are the proteins that tied to each other the endothelial cells, that make that this barrier is so restrictive. That's not happening in the rest of the body. So the main cells involved in the blood-brain barrier are the pericytes, the astrocytes, and also the shear stress, because the fluid that we have here of blood is making that these endothelial cells have a specific shape and a specific production of tight junctions, because also this flow is helping on the production of this tight junction and in the increasing of this restriction in the barrier. So it's important to have all these points into account in our model. And it's also important to know that the integrity of this barrier is affected in many neurodegenerative diseases like Alzheimer, Parkinson, Huntington. So it's important also that it's important to model this barrier in order to understand these diseases. Okay, that's the design that we have done for this blood-brain barrier on a chip. So this is a microfluidic chip with 3 chambers, so 2 lateral chambers that will mimic the blood, and a central chamber that will mimic the brain. And these chambers are separated by these columns, these stacks that has a distance between the stacks of 100 micrometers. So in the central chamber, we can inject a hydrogel that will contain the pericytes and astrocytes and will mimic the brain area of the blood-brain barrier. And then in the lateral channels, we are going to inject the endothelial cells for growing in a 3-dimensional structure, the endothelial barrier. Then when we will have the barrier created, we are going to test the permeability of certain drugs focused in Alzheimer's disease. We're fabricating our nanochannels and our microchannels with polymethylsiloxane by photolithography with a mask. And also we are integrating some electrodes. So this is the design for the microchannels, and this is the design of the electrodes. The functionality of the electrodes is basically for measuring. So here are the stacks that I commented. Here is the brain area where we have the pericytes and astrocytes, and here is growing the endothelial barrier between these stacks that has a 100 micrometers of distance. And for analyzing the creation of this barrier, we are also fabricating electrodes in each side of this barrier. So we have here an electrode, another electrode, and we are passing a current between the 2 electrodes for measuring the impedance, the resistance of this barrier, of this BBB, against the pass of the current. This resistance will tell us which is the stage in the formation of this barrier. So we can analyze which is the permeability of the blood-brain barrier and by the injection of this current measured by impedance spectroscopy. So here you can see the integration of the microchannel. That is a micro integration with a micro cutting and micro masking for putting in a micrometric orientation both the pattern of the electrodes and the pattern of the fabricated microchannels. Okay, here is the formation of this. Once we have fabricated already the chip, we're seeding this chip following these steps. So we start with the brain area, where we are seeding in the central chamber this hydrogel combined with pericytes and astrocytes. And after we left to seeding to grow this culture with a couple of days, and after these 2 days, we are sitting in the lateral channels, the endothelial cells. Then we left to grow this endothelial barrier for 7 days. And after 7 days, we characterized the system, and we fixed the chip. And then we are doing immunostaining for labeling the nucleus, the different cells that we are growing into the system, and also the tight junctions. Okay, here you can see with bright field the stacks that I commented. Here is the endothelial cells that has been growth in between the stacks, and here you have the brain area, and also with electrodes in between the barrier. So here you can see life and death test. We conducted life and death analysis for analysis the cytotoxicity of the chip. And in green, we can analyze the live cells into the chip, and in red, the dead cells. So as you can appreciate, there was not or very little negligible amount of dead cells, and has a good healthy our culture. And also we was staining the nucleus in blue and the tight junctions in between the endothelial cells with the CO1 and the VA Catherine. And as you can appreciate, there is a well construction of these tight junctions in between the endothelial cells. So it's this orange, so here red color, so here with a merge of the colors in orange, the formation of these tight junctions. So once we have the blood-brain barrier in a chip created, we analyze the permeability of this barrier. We have 2 different technologies with electrodes, as I commented before, with this impedance, measuring the resistance through this barrier. Here with this Nyquist plot, you can see the evolution of the impedance. So here we have just the hydrogel without endothelial cells, and when we are passing the days and the barrier is but created, we are increasing the resistance that is in this X is represented the resistance. You can appreciate that the resistance is increasing. This is the model that we use for fitting these impedance results. And also we analyze the permeability of the chip with fluorescence. So basically we are injecting different size of fluorescent molecules in the blood channel, and we analyze after 1 hour the permeability of this fluorescence molecule of different sizes. So smaller molecules are flowing faster, the bigger molecules are, so the small molecules are flowing, are permeable, and the bigger molecules, fluorescent molecules, are not almost permeating after 1 hour. And with this analysis, we can calculate the values of permeability, that is quite well values comparing with the other reported works in this area. And also we have a control without the endothelial barrier that is permeating. It just in 1 minute we can see high permeation. So you can appreciate that the endothelial barrier is working and is blocking the big molecules with fluorescence as the dextran 70. So when we already characterize our blood-brain barrier on a chip, we was ready for using this technology for analyzing different kind of nanodrugs that in this case we had developed in collaboration with the University of Chile with Dr. Marcelo Kogan, in which we generate a nanorods modified with different kind of peptides. These peptides has already proved for blocking the fibril formation, that is one of the main hallmarks described in Alzheimer's disease. So we use this peptide, the D1 peptide that acts for blocking the formation of these fibrils of amyloid. And also we use another peptide that is the angio2 that help in the crossing the barriers. Is a peptide that help in the transitosis of these nano vehicles through the LRP1 receptor. So basically, we have this kind of drugs. Also, we use the PEG with carboxylic molecules for linking a fluorophore for analyzing the permeability of this nano drug, and also a CTBA for giving stability of these nano vehicles. Here in the permeability analysis, in red, you can see the permeability, the good permeability of these drugs after 1 hour. In this case, is with the absence of this angio2, that is the peptide that helps to cross the blood-brain barrier. And in this case, it is with the angio2, so you can appreciate the differences after 1 hour. It's almost double of the permeation of this drug thanks to this peptide. But as you can appreciate, this peptide can cross very efficiently the barrier, and in another paper we have proved the efficiency of this D1 peptide for blocking the formation of these amyloid fibrils. So we had tested in another collaboration other kind of nano drugs based in polymethylene that has been already published, and also with metallate with other natural polymers that are more biocompatible, that has been also published with other collaborations. And as next steps, we're working in increasing the picture of this model, mixing this blood-brain barrier with neurons for having a whole picture of the Alzheimer's disease model into the chip. So we are collaborating with the Laboratory of Stem Cells and Restorative Neurology of Professor Kokaya in Lund University. And also we are integrating biosensors inside the chip for analyzing neurofilament light chains, because it has been discovered as a very excellent biomarker in the extracellular matrix. So we can analyze it in the outlet of our chip, the generation of this neurofilament light that is produced when the neurons is dying, so it's degenerating. So it's quite interesting biomarker for being detected inside the chip. So to conclude, I present the key role of these organ-on-a-chip platforms that could have in the drug discovery and also in modeling different diseases. As you can see, it has been developed for many different kind of organs and for many kind of diseases. We also we had incorporated a sensor based in the analysis of transendothelial resistance to analyze the performance of our blood-brain barrier membrane, the permeability of this barrier. We successfully developed the blood-brain barrier on a chip with integrated permeability sensors. And also we had tested the permeability of nano drugs, in this case nano drugs, but also with other collaborations, we had analyzed different kind of drugs for neurodegenerative diseases. I would like to thank to all the nanobioengineering group in the Institute for Bioengineering of Catalonia, also the Cyber, that is a virtual institute in Spain that is part of our group, also the University of Barcelona, and the Ministry of Science of Spain for funding all this research. And thank you all for your attention. If you have any question, I'm now accessible for any questions. And also here you can see my email address if you have any doubt or if you want to collaborate with this research. So thanks for your attention.